In this video, we'll look at the Identify Restriction Enzyme Sites data function. The Identify Restriction Enzyme Sites data function quickly searches nucleotide sequences for restriction enzyme sites and provides a visualization of the sites that are found relative to the sequence. The DNA sequences used here were imported from a text file and the column set to a sequence type. From the data function settings dialog, enter the data table and the column containing the sequences to be searched. By default, a common set of enzymes is selected, which includes enzymes that are known to be available from commercial suppliers. Individual enzymes can also be selected by clicking the checkbox to the left of the enzyme name. The list of enzymes is very long, but you can search for specific enzyme names by typing their name into the selection box. For this demo, I'll select a few enzymes that I'm familiar with from my days in the lab: EcoR1, BAMH1, BGL2, and SMAW1. When the data function is finished, a new column is created that contains the original sequence searched and annotations for each enzyme that was found. Below the sequence, a gray box indicates the start site of a restriction enzyme site. Hovering over the annotation provides information about the restriction enzyme site, such as the enzyme's target sequence, its top and bottom strand cut sites, the strand containing the 5' to 3' sequence, and location within the sequence. The column containing the sequence and the annotations are stored in a GenBank format and can be exported from ChemChart's table visual. To copy the GenBank file for an individual cell, right-click in the cell and select Copy from the pop-up. Under Cell, select Text. This will copy the GenBank file for this cell to the clipboard, which can then be pasted into a text editor. The enzyme site details that were shown in the annotation pop-up are stored as features in the GenBank file. To export a GenBank file for an entire column or for a subset of cells within a column, select Export from the pop-up and then choose GenBank as the export file format. Exported rows can consist of all rows or only the marked or filtered rows. GenBank column will be automatically filled with the column which was right-clicked, but this can be changed to another GenBank column if needed. IDs in the GenBank file can be specified from an ID column. Clicking Export lets you specify the file name and location for the exported file. Thank you for watching, and check out our other lead discovery ChemCharts tutorials.